Welcome to the video to walk you guys through Unit 2, Investigation 3, Evidence 1, Lab, Diffusion through a Membrane. Now we're going to review the introduction or the background. Molecules are constantly moving. They move in straight lines unless they are deflected by other molecules or obstacles in their environment. Diffusion is the process by which the collisions between molecules cause them to continually spread apart from each other. Their overall movement can therefore be described as movement from an area of greater concentration to an area of lower concentration. We call that moving down the concentration gradient. Diffusion continues until the molecules are equally distributed. That is, their concentration is equal throughout the area that contains them. We call that equilibrium. At this point, the molecules continue to move and collide, but their concentration remains the same throughout the area of containment. When certain molecules encounter artificial membranes with pores, they may be able to pass through. If the molecules are small enough to pass through the pores, their movements eventually will cause the concentration of these molecules inside and outside the membrane to equalize. Living cells are surrounded by a membrane that acts as a selective barrier between the contents of the cell and its environment. The membrane is selectively permeable. That means that it allows some molecules and other particles to enter and exit while it blocks others. The permeability of the membrane can change depending on changes in the internal or external environment of the cell. As part of this activity, you will build a model cell using an artificial membrane. Remember that this membrane is only a model. Unlike a cell membrane, it will always have the same permeability to dissolve substances. Small molecules and water will be able to pass through easily, while larger molecules will not. Our objectives for the lab are as follows: to demonstrate how to test for simple sugars and starch using chemical indicators, to explain diffusion through a membrane, and to describe the permeability of a model membrane for glucose, starch, and starch indicator solution. The materials we used for this lab are as follows: dialysis tubing, string or unwaxed dental floss, we used dental floss, glucose indicator solution, it's also known as Benedict's solution, a test tube rack, concentrated glucose solution, tap water, 8 test tubes, starch solution, paper towels, droppers, safety goggles, starch indicator solution, which is iodine, a 250 milliliter beaker, a hot water bath, and a test tube holder. Okay, so before we get started with the procedure of the lab, I want to talk a little bit about what dialysis tubing is. It's kind of like plasticky. You can hear it being all crinkly. Dialysis tubing is something they use in dialysis. When your kidneys don't function right, the purpose of your kidneys is to filter harmful things out of your blood so you can, pee it out. When your kidneys don't work, you still need to get rid of those toxins and the waste. So they put you on dialysis, where they actually take your blood out of your body and filter it through a machine. And dialysis tubing is used in that way. Dialysis tubing has really, you can't see them because they're itty bitty, teeny tiny little pores that allow the stuff to pass through the tube. So it's essentially a selectively permeable membrane. It allows certain things to pass through, but not others. So that's what we're going to wind up using. And the first thing we have to do before we do anything else, according to your lab, is to make a cell. So we're going to make a dialysis tubing cell. So in order to make a dialysis tube cell, the first thing you have to do is soak it in a little bit of water so it's not so crispy. So you want this to be leak proof. So what we do, right, we take it, it works with a partner, so Mr. England's helping me out. We take it, we fold it in half, we crease it, we twist, and then we tie. So Mr. England is going to tie this bad boy. Oops.
If I don't break something.
I was thinking the same thing. All right. And when we do this, if this was an actual lab that you guys were doing, one thing that would happen is, you guys, this would be the hardest thing for you guys. We make it look easy because we've done it like a million times before. See, I would totally keep tying that, but on the other side and around.
Yes, boss. I don't know.
Because I have like issues when it comes to
If it leaks.
this leaking. Right, exactly. We definitely don't want this to leak, but your hands are stronger than mine. One more, I think we're good.
Hopefully.
All right, there we go. Now, in order to open it, it's this magic thing. You turn on the water, and if you rub it between your fingers when it's a little wet, the tube should open. Come on, tube. Ha ha ha. Maybe. Yep, I got it. It's another thing that kids always have a hard time with is, thank you, the tube. Okay? So now we've got the tube open a little bit. My finger can go in there. So we're going to put the two solutions in it. We're going to put a little starch solution. The starch solution is white. Can you give that a stir, Mr. England? Even though it's supposed to be soluble, like dissolvable in water, it's not very good at it. It's not very good. Okay.
That's what you get from a large polysaccharide.
Yes. So we're going to put a couple squirts in there. Helps if you stick the pipette all the way down in there. Oops. If I was feeling brave, I would just dump it in through the spout of the beaker, but the mess could be epic. Give me one more so we have enough space. Maybe half. I'll do half. All right. So there's the starch solution, and this one's a glucose solution. I did 3.5, so I'll do 3.5. So we have consistency. There you go. Should I do a little more?
No,
We're
it's
good?
all right. It's all
Okay. So we end, let me get out the air. We close it the same way we close the other end. Fold, crease, twist, pinch, and tie.
The strings up.
Yep, I was just getting ready to say.
They drive me crazy.
I know, they're way too long, but we need the longer floss. Yeah, we're just using dental floss to tie. We need longer floss to make it easy to tie. It's what happens when you get dollar store dental floss. All right, and the lab now says we need to rinse this.
To make sure there's nothing on the outside.
Mm-hmm. We're gonna pat it dry. Can you grab a paper towel, Mr. England?
Yep.
Pat it, not squeeze it, because we don't want our work to go awry. Ooh, be careful. And now we're going to get its mass. So now we're going to get the mass of the tube before we put it in. 15.4 grams. You need to record that on your lab. The mass is 15.4 grams before. On page 3, number 6, it's at the top of the page, it says record the mass of the cell here. That is where you put the mass of 15.4 grams. So now the lab says to put the cell in a beaker. So here's our beaker. We're going to gently place our cell in the beaker. Smush it down in there. And cover it with water. Just regular tap water works. Okay, it's covered. And it says to put the starch indicator solution, which is iodine, into the beaker. And I need to add enough to make the water an amber color. That's what iodine looks like. You know what? There you go. There wasn't much in the bottle anyway. So it says to label the initial state part of the diagram found on page 5. So I think so you need to indicate the contents of this beaker and the tube and the color of it. So we'll snap a picture in here after we're done, after this part of the video, so you have an idea of
what to put where.
These two images are two views of the initial setup. You need to label the contents and note the colors present in the beaker and the cell in the initial state diagram, which looks like what you see below. You can abbreviate iodine with an I, glucose with a G, starch with an S, and water as a W or H2O.
So now we're going to do part 2, the chemical testing. When we do part 2, we are establishing controls. So you guys can see what positive and negative tests for these macromolecules looks like, okay? So when we test for a starch solution, we use the starch indicator solution, which is the iodine. You can't tell, we looked at this before, but it's amber. All right. And when we test for glucose or any other monosaccharide, we use Benedict's solution. Now, Benedict's solution is in the brown bottle, but if I do this, it magically is this really pretty blue. I'll move this way so you can see it from before. Okay. It's this really pretty blue color. All right. So if we look at the directions, it says to get 6 clean test tubes. Voila, here they are. And we're going to test distilled water, starch, and glucose with each of the 2 solutions. So I'm going to make this row, column for you guys. This is going to be water, this is going to be the glucose, and then this is going to be the starch. So I'm going to take a pipette. It says 10 drops. A pipette is about 10 drops. So this is our water. All right. Then I said glucose next. I'm going to put glucose right next to it. And then I'm going to stir the starch up a little bit. And then I'm going to put a pipette full of starch in the last tube. All right. I'm going to label these. Water, glucose, starch, so I can tell them apart. Water, glucose, starch. Okay. If we do the test with the Benedict's solution, all right, the way the Benedict's solution works, that's the right one. We have to put it in a hot water bath. So I'm going to do the Benedict's test first. I'm going to put a little pipette of Benedict's solution in the water, in the glucose solution, and in the starch solution. Close that back up. They have to sit and boil in a hot water bath to allow the reaction to take place. So there they are. And I'm going to go put them in the hot water bath. Ooh, there's more.
Okay.
There we go. Ooh. Okay, so they're going to sit for a couple minutes. And while those sit, we can do the starch indicator test. All right. So iodine, like I said before, is our starch indicator. You saw it when we poured it outside the tube. Okay, so I'm going to put pipette worth in here. Okay, it's still the same amber color. I'm going to put another pipette in the glucose solution. It is still an amber color. See what happens when we put the pipette in the starch tube. Okay, I'll move this way. It changes to this inky black color. It's almost purple. Okay. So if we take a look at these, I'm going to pick them up. We've got the water, the glucose, and the starch solution. All right. The test results that you see here for the water and the glucose are negative. So that means when you put the starch indicator solution in with water and a glucose solution, there's no starch, so they're this ambery color, all right? When you put them in, when you put starch indicator solution into a starch solution, it turns this blackish purple inky color. This is a positive test. Okay, so in your table, when you look at the controls, table 2, the chemical test results, it's negative for water, negative for glucose, but positive for starch. So when iodine and starch get together, this is what the result looks like. Okay? All right.
This picture shows you a close-up of the results of the chemical testing with the iodine solution in water, glucose, and starch.
We can take a look at the test results for the glucose solutions. Hopefully this is going to work.
Good.
Good. So there's the test for water. Okay, I'm going to put that down. My lab skills are still strong. Here's the test for glucose. Okay, it's that orangey rusty color. And here's the test for starch. It looks a little milkier, but it's still that blue color. Okay. So now if we fill in the top part of table 2 for the blue colored glucose indicator solution, we need to figure out what's a positive test and what's a negative test, all right? So the solution in water is still that blue color. So this is going to be a negative test for glucose. All right, so the water is a negative test. The glucose, when you put the glucose indicator solution in a glucose solution and you boil it for a few minutes, you heat it in a warm water bath, it's going to turn this orange color. This is a positive result. Okay, so when you put Benedict's solution, the glucose indicator solution, in with glucose, it's going to turn this orangey color. And then the starch test is still that same blue color of the Benedict's, so that's going to be negative.
This picture shows a close-up of the results of the chemical testing with the Benedict's solution, or the glucose indicator solution, in water, glucose, and starch. Here you see a picture of the results of both sets of chemical tests. The Benedict's tests are on the left, and the iodine tests are on the right. The table you see summarizes the results of the chemical testing you should have recorded on Table 2, which is found on page 4 of your lab.
So we have let it sit, and we're going to do the post-lab stuff. So it says record the mass of the cell after letting it sit. First of all, take a look at it. Some things have changed. It was not purple, black when we put it in. Okay, so take a look at that for a second. Okay. All right, so now let's get the mass. I'm gonna grab it. Gently pat it dry again. Thank you, Mr. England. There it is. And I'm going to put it on. Has a mass of 16 grams, 16.1. We'll call it 16.1 grams. Okay, so the mass has definitely changed. So now we have to do the chemical tests that we did in the previous section to the liquid outside the cell, the liquid that was in the beaker. So I have one little test tube of the material. I'll finish this one out, just a pipette. Give it a fill, okay? One we have to test with the Benedict solution, the blue stuff, to look for the presence of glucose. And then the other we're gonna test with the iodine solution to see for the presence of starch. So take this one, there's that blue iodine, Benedict solution. We're gonna put it in, and we're gonna let it sit in our hot water bath for a few minutes. There we go. And then we will test this with the iodine solution to see if the starch moved. Actually, we don't have to. We're just doing the glucose test.
Because it's what turned
Yep.
purple.
So one way you can tell about the movement of starch is the color of what's in this beaker. So the color of the water outside the beaker is pretty significant. Can you see it changing?
Mm-hmm.
We have some interesting things going on.
Mm-hmm.
It's like magic before your eyes. I don't even think we need to wait 5 minutes
No.
to see what's going on here. Okay, so this was the liquid outside the test, outside the cell, outside the dialysis tube, tested with the glucose indicator, the Benedict solution. You have to go back to the previous section to table 2 to determine whether that is a positive test for glucose or a negative test for glucose.
Here are 2 close-up pictures of the setup after we let it sit. The inside of the tube is a purplish black, and the outside of the tube is a lighter yellow color. At this point, you need to take the results of the glucose test and the observations of the fluid both inside and outside the tube and determine what molecules moved where. You will do this by completing the final state diagram on page 5 of your lab and answering the questions. In your analysis, you need to address the motion of all 4 molecules. As you analyze the results, as a reminder, those molecules are glucose, starch, iodine, and water. As you answer the questions, you need to describe how these molecules moved and what evidence you have to support that claim. Remember, if you are stuck on any questions, it might be a good idea to refer back to the background information for the lab.